On our journey to be a pathology superhero, we're going to learn today about upper gastrointestinal pathology. The key concepts in upper GI pathology are that Barrett's esophagus is intestinal metaplasia caused by acid reflux. It's a precursor to cancer. Helicobacter causes gastritis and intestinal metaplasia. It's also a precursor to cancer. Peptic ulcers have a different pathogenesis in the stomach and duodenum. This is something a pathology superhero knows. Celiac disease is caused by an immune reaction to gluten and results in malabsorption. And the fifth key concept is that carcinoid tumours can be benign or malignant and give rise to carcinoid syndrome. Let's explore these key concepts a bit more. Reflux esophagitis is caused by an incompetent lower esophageal sphincter. As we can see here, acid refluxes back into the esophagus, and this can affect the epithelium lining the lower esophagus. The acid in the lower esophagus can cause ulceration, and the ulceration can heal by fibrosis, giving rise to strictures. More importantly, and less treatable, it can give rise to carcinoma. It does this in the sequence illustrated to the right. A normal esophagus subject to acid will undergo intestinal metaplasia and produce Barrett's esophagus. Further reflux and inflammation will then lead this benign epithelium to accumulate mutations and then develop low-grade and then high-grade dysplasia. Further mutations will lead to the development of adenocarcinoma. So in summary, chronic reflux leads to metaplasia of the glandular epithelium at the base of the esophagus into gastric or intestinal type epithelium. Squamous cell carcinoma also occurs in the esophagus, whereas adenocarcinoma occurs in the lower esophagus where there's acid reflux. Squamous cell carcinoma more commonly occurs in the middle part of the esophagus. It starts as a grey-white thickening of the mucosa, carcinoma in situ, and then it can grow into the underlying mucosa and become invasive squamous cell carcinoma. It can appear polypoid, fungating, an ulcer, or diffuse infiltration. It looks quite similar to squamous epithelium. It looks quite similar to squamous cell carcinoma at other sites, with dysplastic cells with intercellular bridges and the production of keratin. Let's move on to think about the pathology of gastritis, gastric ulcer, and gastric tumours. They're all linked by one thing. And that thing is Helicobacter. So Helicobacter is a commonly acquired bacteria. It's acquired through drinking contaminated water. In the Western world, we often acquire it as adults, but in the developing world, children acquire Helicobacter. It's a curved gram-negative organism that we can see in the gastric mucus, and as it lives there, it excites an antigenic reaction and releases enzymes that dissolve the mucus lining the stomach. So, how is it associated with duodenal ulcer, dysplasia, and gastric cancer? Here we can see a picture of Helicobacter. It's those dark blue seagull-like organisms that are in the center, in the lumen of this gland. Helicobacter causes about 60% of antral gastritis. That's gastritis in the lower part of the stomach. Another common cause is bile reflux, as the sphincter at the bottom of the stomach becomes incompetent. What we see is a loss of gastric folds. It causes atrophy, a shiny glazed mucosa. So what do we see down the microscope in cases of Helicobacter infection? What we see is chronic atrophic gastritis. This is a mixed chronic inflammatory infiltrate throughout the mucosa and includes lymphoid follicles. There is loss of glands and intestinal metaplasia of the remaining glands. This is really important to remember. There can be some neutrophils in the epithelium, and we'll always see the Helicobacter, these small spirochetes in the centre of the glands. The reason why intestinal metaplasia is important is because, like in the esophagus, it is a precursor to cancer. Once intestinal metaplasia has occurred, mutations accumulate and the cells become dysplastic. Further mutations lead to the ability to become invasive, and we have gastric carcinoma. The picture at the right here shows the goblet cells of intestinal metaplasia at the bottom right, and it shows the pleomorphic, hyperchromatic, mitotically active dysplastic cells that are now in the gastric epithelium. What does gastric cancer look like? There are two main kinds. The first is intestinal type, where we have malignant glands infiltrating through the stomach wall. The second type is the diffuse type. Here they have individually infiltrating cells, signet ring cells. These cells have lost the E-cadherin gene, the Velcro that sticks cells together. Macroscopically, we describe this as Linitis plastica, or leather bottle stomach. Let's move on to thinking about peptic ulcers and their relationship to gastritis and Helicobacter. Peptic ulcers occur in the stomach and in the duodenum, and the pathogenesis of each is quite different. Gastric ulcers have decreased mucosal resistance and low acid levels. So Helicobacter will be associated with gastric ulcers because their enzymes, the ureases, attack the mucus that protects the gastric epithelium. So once this mucus is gone, then the gastric ulcer can occur. Duodenal ulcers are due to excess acid and pepsin. They're due to dumping. They're due to dumping of gastric acid in the proximal duodenum. They can be associated with Helicobacter, but also with many other conditions that increase acid production. Let's move on to thinking about celiac disease. This is an immune reaction to gliadin, a protein in dietary gluten. Anti-gliadin antibodies can be found in the mucosa of the duodenum and also circulating in the blood, which means that we can do blood tests for celiac disease. Celiac disease tends to affect women more than men, and in particular, it affects people with a certain HLA type. HLA, human leukocyte antigen. This is all about the self-recognition system in the body. And people who have certain types of HLA are much more likely to get celiac disease. In fact, it's hardly ever found in people who don't have these HLA types. Diagnosis depends on blood tests, excluding other causes, and improvement with a gluten-free diet. Sometimes, however, people with celiac disease need a biopsy to make a diagnosis. This is what a normal duodenum looks like. Can you see the long finger-like villi? These increase the intestinal surface needed for absorption. In celiac disease, the immune reaction means that these villi are all gone. The surface is now flat, so there's much less area for absorption. That's why people with celiac disease get malabsorption. Our final topic in the upper GI tract is carcinoid tumours. So carcinoid tumours can occur widely throughout the GI tract, and they can also occur in the lung, but most commonly they occur in the appendix. It's usually discovered incidentally as a bulbous swelling at the tip of the appendix that tends to spread through the wall of the appendix. Now, most tumours will metastasize to lymph nodes, and carcinoid tumour is no exception. But in the appendix, it hardly ever metastasizes. Carcinoid tumours in the stomach, however, metastasize frequently and early. So the outcome of carcinoid tumour doesn't depend on grade or stage as much as position in the GI tract. Carcinoid tumours are an example of what we call well-differentiated neuroendocrine tumours. Neuroendocrine tumours grade 1. There are high-grade neuroendocrine tumours. We'll learn about some of them in the lung videos. Small cell carcinoma of the lung, for example. On our journey to be a pathology superhero, today we've learned a lot about how cancer occurs in the upper GI tract. Metaplasia leads to dysplasia, leads to carcinoma in both the esophagus and in the stomach. We've learned about how Helicobacter contributes to this, as well as peptic ulceration. We've learned about celiac disease and how it's restricted to people with certain HLA types. And we've learned about carcinoid tumours, neuroendocrine tumours of the bowel. We're well on our way to becoming an upper GI pathology superhero.